Okay, let's see how this works. This is the most common errors we see on the lab report video. I think it's really important as you do your lab report, you understand these things and make sure that you do not make those same errors. Um, just kind of go through the list and explain. You'll see a lot of these again and again in each section. Also remember that the 3 sections you're actually turning in for credit are the introduction, the results, and the discussion. So those are the only graded, those are the only ones that should be there. So we'll look at the introduction first. The first point is improper heading. The header should be left justified, all caps, bold. It should look exactly like this right here. Right? So part of your grade is, can you follow directions? No underlining, no colons, not centered, not small letters. It should look exactly like what I circled. And that's true for every one of these. Introduction, the results, the discussion section should all be that format. One of the confusing parts that sometimes students have is they have too few reference or a complete lack of references. Even though I am not grading your reference list, your bibliography, whatever is the works cited, whatever section you'd call it, you need to associate the introduction with scientific principles that would be supported with a reference, right? So we need to support with a reference here in terms of your scientific principles. There's a number of different ways you can do it. It really doesn't matter the format, but oftentimes in science, the 2 most important components of a reference are who said it and when did they say it. So after citing something or taking information, or another way to think about it is place blame on somebody, then cite the last name and then the year in parentheses. If you don't have a year, it would be the date accessed, so it'd be probably 2020 as an example. If you don't have a name, then you could put anonymous. And if you had multiple ones, you'd have anonymous A, B, C, D, etc. And that's how they would appear in your reference list. But again, we're not doing a reference list, but I do want you to demonstrate you know how to reference information in a scientific paper. One of the problems we see sometimes with scientific papers, that's the third bullet, is students are taught, especially in English courses, to ask rhetorical type questions. In science, we want to get straight to the point. We don't want wasted space, and we just want to give the idea without the question. When you're writing your introduction, you should also give a general overview of the experiment. So you should basically give it just a quick synopsis of what we're doing. Make sure to focus on the important pertinent facts in terms of the information regarding your study. You don't have to do it, but it helps if you put it in some sort of historical context. If it's not historical, it needs to be practical. So you need some sort of context. If you have a very practical paper, then you don't need to put the historical stuff, but it helps to give perspective. So the whole idea is to give perspective for that. You should briefly discuss what your experiment is. This is what we did. This is how we did it. Right? And this is what we expect. So, we tested the water bath, looking at the negative feedback controls with it by placing water cold water in the water bath after it reached temperature, etc. So you should always have your expectations in there, right? So you should have some expectations as well. Be careful of spelling and grammar. This is a college level science paper. The expectation is it's well written. Common errors are misspelled words, disagreement of tenses, incomplete sentences, things like that. Proofread your work. This is a good thing to remember. Make sure to finish the paper in plenty of time to proofread it. If you can, let the paper just sit for a couple days, and then you might pick up things you otherwise might miss. An even better strategy is to have someone else read it. but I am likely probably the only other person that will read it. Another thing you can do, which seems to help, is change the font or the color of the font. Make it different than what it is, and oftentimes when you reread it, then you'll pick up more stuff. So I would save it and then like select all, make it, if it's in Arial, make it Times New Roman or vice versa, and then make it a different color that's easily read, but that helps too in terms of that. Continuing on, you should discuss the significance of what you're doing. Again, trying to put it in perspective and ask yourself, what's the significance of our study? Sometimes the significance is just being able to measure glucose so that we can tell if you have diabetes. And sometimes it's to demonstrate a principle. We're showing negative feedback or heart rate stuff, whatever it is in terms of that. You should always make sure you have purpose and objectives with a hypothesis. One of the things that we practiced a little bit in the JLR, so you should have that, is what's the hypothesis for your experiment. Remember, if you use the if, then, because statement, if we exercise after blowing through a straw to change the color of the solution, then we expect it to be faster after exercise because when you exercise, you produce more CO2, which changes the acidity of the solution faster, so we should get a faster color change. That's the type of thing that you would want for that. In a paper, you should always spell out abbreviations the first time in each section. You should spell out even common abbreviations. In fact, as an example, most people would understand if you just wrote DNA, but if you say deoxyribonucleic acid, then some people go, huh? Oh, DNA, okay. But once you've done it once in a section, you can use abbreviation again and again and again. One of the more common errors I see is capitalization. Just be careful of it. Many students want to capitalize words that aren't capitalized. Amino acids and carbohydrates are a common one. If you say, deoxyribonucleic acid, in parentheses DNA, even though the DNA is capitalized, the word deoxyribonucleic and then the word acid is not. So, proper names would be capitalized. Obviously, beginning of sentences would be capitalized. Diseases usually aren't capitalized unless it's named after somebody. So diabetes mellitus would not be capitalized, but Crohn's disease would, as an example. So just be careful of capitalization. Here's a good thing. If you can do it, say what you mean what you say, right? So select your words carefully and make sure they have the intended meaning. Reread your papers to make sure they're relaying the information that you want. In laboratory, especially with one experiment we typically do, we never prove anything. We show things, we demonstrate principles, but we never prove anything. In fact, in science, you really do very rarely prove anything. So be careful of that. Include all elements of the exercise. Make sure all elements of the laboratory are presented. General rule is that if we did it in class, then you are responsible for it, even if we didn't personally perform it. And when I say the exercise, I mean, FOX 1.3a or FOX 1.3b, not both. FOX 8.4a or b or c, or FOX 2.1a or b or c. You don't have to do all three of those, the ABC components, but you should answer about them. So the easiest place to think about that would be in the heart rate one, where we looked at the individual heart rates. You probably should talk about that a little bit. And then we looked at the class average, and you probably should mention that as well. So that's like there's two parts to that one. The other one is make sure it's the appropriate length. The proper length depends on the writing styles, the subject matter, what you decide to present, and things like that. But generally, at least 1 page in length to cover the scope and background is needed. If you start to go over 2 pages, it's probably becoming too detailed. It's not that the information isn't necessary, and almost always is, but it's not the place you necessarily want to present it. You can just put that in the discussion. So once you start to get to that third page for the introduction, it's probably becoming less of an introduction. So keep an eye on that as well. But, spilling over to a second page and ending there somewhere on that second page is probably a good rule of thumb. Now we'll move on to the results section, which again, so here's our results, right? One of the common errors that students have is they don't have the proper heading. So again, it should look like this: all capitals, left justified, bold. When we get to discussion, it'll be the exact same thing as was the introduction. For the results, one of the biggest problems students have is they don't have both a text section and a visual explanation section. So you should have a text section at the beginning. That's words that describe the table. So your text section needs to describe the data. So if you were to make a video of it just like I am and describing the data to somebody, what would you say? That's what you should have in terms of your text section, to describe the tables and figures and the data in your report. The idea is to make it come alive for the reader by describing it. So that's the first section. That's a text section. The second section should be tables and figures. Okay, so the tables and figures should come after the text. They don't have to be consecutively. You can put the text on a separate page in the tables and figures. Sometimes it's safer to put tables and figures in individually as well. It depends. You can hand draw them and submit them like we kind of practice with the PDF file in terms of your report, and do that. You can do them on a computer. We'll talk about that a little bit as we go through, but you need tables and figures as well. When we name tables and figures or label them, there's a couple things that we need to do properly, and we'll get there as we move down. So you need titles for your tables and figures. They should be titled properly. In a scientific paper, you may only have tables and figures. So basically, when you look at a table, it's rows and columns of things. Okay, so I'm going to erase this so I can see. As a matter of fact, I'll make this a little not quite as wide. When we look at a table, just go, is it rows and columns of things? If it is, then you call it a table. If it's not rows and columns, then it is a figure. So those are the only two things you can have. So you don't have a chart or a graph or a map or a picture. Those are all called a figure. Okay, so the two things you have is tables or figures. Essentially, anything that's not made up of rows and columns should be a figure and needs to be labeled that way. Okay, so when we have figures and tables, we need to label them consecutively. Okay, so tables and figures are measured independently throughout the paper. So if you decide you're going to do glucose in your introduction, you think it's important to have a diagram of glucose, like a Fisher projection of glucose, what it looks like, then you would call that figure 1, because it's not a table, it's not rows and columns, so it'd be figure 1. You would have a description of that, a title, and then make sure that you describe it to the reader so they can just look at that without having to refer to text. And then you stole that from somewhere, so you probably should cite it. Right? If you got off the internet, that stuff's easy to find on the internet. Just go to, glucose and then Google images. You'll find a ton of them. You need to reference those as well. If you have then another figure in your results section, that would be figure 2. And then if you had a table, that would be table 1, because they're measured independently. You just follow this pattern throughout the report. Again, figures and tables need to be properly labeled. Okay, so label them properly. They only have a single designation: figure 1, figure 2, table 1, etc. You don't call them figure 1A or figure 1.1. It would just be figure 1, figure 2. Here's the thing that really becomes important is figures and tables stand on their own. I alluded to that a minute ago, and what stand on their own means is that they have sufficient information on the actual table and figure information that you don't need any other reference material. So they should have a number designation, so table 1. They should have a title, what it shows, and they should have a description. That description is usually a sentence or often sentences. And kind of think about it this way. If you were walking down the hall and that just fell out of your backpack and landed in front of the anatomy room, and some student who has never had physiology picked it up and looked at it, they would be able to figure out what it was, even though they've never done the experiment. So stand on their own, very important concept. Another small but often mistaken thing is that the spacing and font and everything for the paper. We say the paper in general is double spaced, normal font, normal margin. So 10 or 12, and use some sort of normal font that's easy to read, like Arial or Times New Roman or something like that. But the figures and table titles should be single spaced. Okay, and that's going to distinguish it from the text. All right, and oftentimes the figure or table information will be very similar to what the text says, because you're explaining it and making it stand on its own. So you need to explain it a couple places. So here's a really good place to bring this up. If you don't feel like you're being redundant when you write this paper, you probably haven't done it right. So one of the problems students often feel they have when they're writing the lab report is the idea of redundancy. They feel that they have said it already, and why do they need to restate it? In English and other classes, you would actually get marked down for being redundant. In science, you would be marked down for not being redundant. And the idea is, when scientists read scientific papers, they don't typically read them from cover to cover. They don't read the entire scientific report at one time. And so oftentimes when I was doing my research for my doctoral dissertation, I had literally 500 journal articles I had to read. I did not want to read every portion of every one, so I just flipped to the results section or the discussion section or the method section. How did they do it? And they needed to have that information that I needed there, so I didn't have to flip through. So that's why scientific information tends to be very redundant. We'll see this especially in the discussion. But in terms of the results, you're going to have what seems like on your figures and tables very similar information to your text. That's actually a good thing. Don't worry about it. Worry about it if it doesn't seem like that. Okay, but the point of all that was to distinguish it from text, you need to single space your figure and table titles. It's not on here, so I'm going to mention it now anyway. I don't care if you do your tables and figures by hand or on a computer. To be honest, doing the tables on a computer is really easy, and I could show you how to do tables very simply, and you can make some very nice tables in a very short amount of time. For the figure, though, if you want to draw a figure, it's a little more difficult, sometimes a lot more difficult, and many students end up, either making the outline for the figure on using a computer and then draw the lines or the chart in by hand, or do the whole thing by hand. And oftentimes students that have tried to do it on a computer, after getting frustrated literally with an hour or 2 or 3 of work, decide I'm just going to do it by hand. The advantages of doing it by a computer is if you have to make a change, you don't have to redo the whole thing. But the disadvantage is it's not a very good, easy learning curve, and it might take you hours to do something where you could have drawn by hand in literally 1/10 of the time in some cases. And I'm not going to give you any extra credit one way or the other in terms of doing that figure. As long as it shows what you want it to show, you should be fine. Once figures and tables have a name designation, you're going to capitalize that. So if we say in general, here's a figure or a table, that's not capitalized. But once you give it a specific number, like figure 1, now it's got a proper name and it needs to be capitalized. So you would say figure 1 would be capitalized, or table 1 or whatever. The other thing we see sometimes in the results is do not discuss your results in the results. So you present the data, but you don't say, oh, somebody, you could say, oh, Joe's blood glucose level fasting was measured at 137. This is indicative of diabetes mellitus. That's fine, but you wouldn't explain diabetes mellitus or any of the other things with it. That's saved for the discussion section, which we're moving on to next. Okay, so here's your discussion, and I don't grade on a specific rubric for this paper, right? So remember, the lab report is 50 points total, and I don't say, oh, the introduction's worth this many, and the results is worth this many, and the discussion is worth this many. But if I did, at least half, and probably closer to 2/3 of the total points would be in your discussion. Your discussion is the most important part of the paper by far. I didn't mention length a whole lot in terms of the results, but if you go back to the results, most people's text are less than a page, and in some cases, like a few sentences. Like if you did the hyperventilation one or the blowing to the straw, changing the pink to clear one, those would be very short text sections for your results, and you'd probably have one table literally comparing the pre and post numbers. That would be very simple. But if you did something like the blood glucose or the acid-base water buffer experiment, then you're going to have, since there's more components to it, you're going to have a longer text. I don't expect the vast majority of you to have text that's more than a page in any case. And then depending on how many tables or figures that you want to include, then you need to make sure that you just have those that are important. Be careful of putting in redundant data. Don't put in a table and then literally make a figure from that table and put it in. Decide which one's best and just present it that way. So you don't need to do both of those. Matter of fact, you shouldn't do both of those. You should just do one. So if you put if you have one short paragraph and one table, it's going to be pretty short. You might have a whole page, a paragraph, and you might have 2 or 3 tables and figures in results, and you might put them on separate pages. It could be 4 pages, or you could have them all on the same page. It doesn't matter to me, whatever works best for you. But as an example, if you do the heart rate one, I think putting your individual heart rates up as a table is probably not a bad way to go, and probably the class average heart rate is not a bad way to go either. Also, if you don't have good data for the experiment you're doing, you're always welcome to use the sample data presented in the lab supplement, and just consider it yours and use it that way. So with that background, let's take a look at the discussion. Okay, remember, so the discussion, while we're kind of talking about the subject, in terms of length, right? We said the introduction, at least a page, and probably not much more than 2 because you're getting away from the introduction portion. The results I just explained, not much text, less than a page probably for the vast majority of you, and then how many other tables and figures it takes for you. In some cases, you should have, in all cases, you should have at least one table or figure. And in many cases, you'll just have one, but some people will decide to put in other ones to help their explanation and things like that. Your discussion typically is going to be at least 3 pages. I'm not saying 3 pages, I'm saying at least 3 pages. It's hard to put all of the stuff that you need to put into the discussion. Do a good job. Let me know you know what you're talking about in less than 3 pages. I've had some really good ones that were 3 pages, and don't stop when you get to 3 pages. I've said enough. You need to show me you understand the relationships, why we do this, why it's important, what's the physiological associations with it, and things like that. But figure your discussion should be at least 3 pages. I've had discussions literally that were 15 pages long. So you probably don't need to go into that much detail, but just giving you an idea that 3 pages is typically what most people need to do for a good discussion of what they should be looking at. So starting the discussion and looking at it, we've got the two we've had before, the improper heading, right? So it should look like this, all caps, left justified, bold, right? We talked about this for the introduction, right? The references, you should reference this, okay? Again, the exact same idea. So you should give credit, or if you want to, blame for citing those. So if you said, oh, blood glucose, you know, we've said, oh, our person's normal because normal blood glucose is 70 to 99 and our subject was 86. You need to say, where'd you get that number from? And now you can say like Fox, 2016 or whatever year Fox you have in terms of that. Like we saw before, don't ask rhetorical questions. Again, in science, we're just going to go straight to the point. So a couple things that you want to do. You want to discuss your results in light of the physiology involved. So you want to reintroduce your results. So a good rule of thumb is to make sure you've mentioned every figure or table from your results in your discussion. And then based on that result, bring up the associated scientific principles. So you're not going to necessarily present them together. You might kind of tell a story and say, okay, I'm going to talk about this result first, and that's going to lead me to the next. So again, maybe using the heart rate data, you might talk about your individual heart rate and how it was measured, what your average was, and how you got to that. Right? And then what normal is and what we'd expect most people to be, and then talk about what controls average heart rates. And then that would bring up eventually exercise level, and then you can bring up the exercise level from your table 2 or whatever in your results, and that would then, you know, serve as a point of discussion for looking at that data, and was it, you know, what you expected and you know, how you expected it. So bring up the specific significance of your experiment. Okay, why do we do this? Why do we care? Again, what's the reason we're doing it? And the more you can make connections and show me you understand it, the better off you'll be in terms of this. Always good, again, to briefly discuss what the experiment is. You go, I already did all this stuff. Yeah, that's what I said. It should be redundant. It should sound redundant. So again, if you don't feel like you've done all this stuff, then maybe something's missing from a section or two. And so, you know, discussing what your experiment is, it's going to be very similar to the introduction. Don't cut and paste the introduction in, but it's going to be the same ideas for that. Like always, be careful of grammar and spelling. We've mentioned that, so I'm not going to harp on that. Proofread your work, right? So proofread your work as well. Finally, a couple other notes. Discuss the significance of what you're doing. So why do we care? Why do we do it? How do we apply it? Provide scientific background. Who said what? What are the implications of having it done a different way? You know, if you're looking at glucose, cholesterol, or protein, what's the implication of having a high or low protein? What do we need protein or glucose or cholesterol for in the first place? If you're looking at some of the other ones, you know, what's the importance of measuring acid base in the body and pH and how that affects blood, and acidosis and alkalosis, or how does ventilation affect blood pH and things like that, or, you know, how does negative feedback in water bath apply to negative feedback in our body, and, you know, mention how it lacks antagonistic control and how homeostasis is important, and, you know, apply it to those things. So that's the type of stuff you want to do in the discussion, right? You want to have your purposes and objectives with a hypothesis again. Yeah, we did it in an introduction, but you should see the same idea. And, you know, was our hypothesis verified with our experiment, or was it not? Again, as we said before, be careful of capitalization. Say what you mean what you say. That idea of we never prove anything in lab, we show and demonstrate it. Again, include all the components of the exercise. It's the same we saw with the other ones. Spell abbreviations. All right, and again, Think about the appropriate length. The length depends on the writing style, the subject matter, all those other things, but in a general rule of thumb, it should be at least 3 pages in length. I have rarely, if ever, read good discussions that gave a significant amount of information in less than 3 pages. And part of this discussion is for you to demonstrate to me you understand what it means, what's the significance, how it relates, why it's important, and things like that. So not all of you will go down the exact same path, and not all of you will think, even let's say 7 people do glucose. Not all of you are going to go down the exact same path, but certainly you're going to show me, hopefully, why that's important and what you thought was important about it. And basically, it's your opportunity to think about it this way. Just go, hey, James, look how much I know and understand about this. Give me a really good grade on it because I know and understand about it. And I was able to relate that information in an effective way to you as a reader. So that's what we're looking at for those. So that's kind of the important common errors that people make, and don't make them. So I think you should probably go back and look at your paper when it's done and look for that. Don't forget, for the stuff you should include, there's a checklist. That's JLR 13. All right, so that checklist, make sure you look at and literally check things off. Okay, so literally check them off and make sure you have them in your paper before you turn it in. So hopefully this is going to help you in terms of getting a much better score on your paper by avoiding these common errors that often, end up costing students points.